Hello, everyone. Welcome to the second day session of Rising Stars in Plant Sciences, RSPS 2022. My name is Rong Chen, a scientific editor for Molecular Plant. It's my great honor to host the second day session of this great event. And today we will have 5 excellent finalists sharing exciting plant research across different fields. Now let's get started. The first speaker will be Marianne Barberon, who comes from the University of Geneva, Switzerland, and she will talk about the plasticity of root permeability for nutrient acquisition. Please, Marianne. Thank you.
Hello, thank you. Thank you for the introduction, and thank you also for selecting my application for this competition, and mainly also for organizing such a great symposium. So as my title indicates, I work on roots and on nutrient transport. And what is interesting with, I'm sorry, I have a problem changing slides. Okay. What is very interesting with roots is that in terms of function, they can be seen as an inverted gut, where the root periphery and its roots hair can play the role of a surface of absorption that is very functionally similar to the epithelia in animal epithelia. And then we will have some barriers that are formed that are similar in terms of function of the tight junction that we find in animal epithelia. If we look in more detail, so the plant root is organized with concentric cell layers. At the periphery, we have the absorption surface with this famous epidermis and its root hairs that really play the role of a surface of exchange with the soil. Then later, we will have one cell layer that is called the endodermis that form barriers. And really at the center of the root, we will have the vessels that will then transport nutrients and water to the aerial part. This organization allows different uptake scenarios for nutrient and water. There is one pathway that we call the symplastic pathway, where nutrients can be transported from cell to cell through what we call plasmodesmata, that are cytosolic connection. Then there is the apoplastic pathway, where nutrients can be transported in the apoplastic space between cells, and this will be blocked by the presence of barriers in the endodermis that are called the Casparian strips. And then there is a third pathway that is often forgotten, but actually the best characterized so far, that is the coupled transcellular pathway, where nutrients can be transported through influx and efflux carriers that are polarized along root cell layers, allowing a cell-to-cell transport. And this pathway is also affected by the presence of barrier in the endodermis, principally by Suberin lamella that can be deposited in the endodermis. So during my whole research life, I've been studying nutrient transport in root at different scales. So when I was an undergraduate student, I was working on sulfate transport in plants in the University of Montpellier in France. Then later I did my PhD still in Montpellier in France, where I work on iron transport in plants. And I ended up doing a lot of protein trafficking and cell biology in this context. And after this, I moved to Switzerland, where I did my postdoc, where I focused on endodermal barriers. And I'm going to tell you a few words about what I did as a postdoc on this topic. So endodermal barriers, I already mentioned that there are 2 barriers. The first are the Casparian strip. They form rather close to the root tip once endodermis differentiate. These Casparian strips are made of lignin, and what they are doing is that they make an apoplastic barrier, which we can see when we follow the uptake of an apoplastic tracer such as PI. Before Casparian strips are formed, this PI can reach the apoplastic space of all root cell layers, epidermis, cortex, endodermis, and the vasculature. But once the Casparian strips are formed, it's blocked after the cortex. But later in the root, in the endodermis, there is a second barrier that established that corresponds to Suberin lamella. This Suberin is an hydrophobic polymer that coats the periphery of endodermal cell. We can easily visualize it with some stainings for Suberin in entire root. And what Suberin is doing is that Suberin is not doing an apoplastic barrier. What Suberin does, it's a barrier of diffusion or a barrier for the transcellular pathway. So we can see that if we follow the uptake of a cellular tracer that can fluoresce only inside living cells, and before Suberin is deposited, this tracer can reach all root cell layers of epidermis, cortex, endodermis, and the vasculature. But once there is Suberin, its diffusion, its uptake by endodermal cell is blocked. So we have these 2 barriers, very nice. The purpose of my postdoc was to characterize functionally these barriers, especially in the context of nutrition. And this is something that was not so easy to do before because we didn't really have mutants or lines specifically affected for this barrier. So 10 years ago, I moved to Switzerland in a lab where such mutants and lines were uncovered, and I started to characterize the function of these barriers. So in particular, I was working with the Schengen-3 mutant, that is a mutant with no apoplastic barrier. This mutant has interrupted Casparian strip, no barrier. So Schengen is the borderless area in Europe, so comes the name of Schengen-3 mutant, so mutant specific for Casparian strip. And then I also work a lot with a line that we develop where we can degrade Suberin specifically in the endodermis, called the CDEF line, where there is no Suberin and no barrier for the transcellular transport. So I did a lot of physiological experiments with the lines. It was quite surprising to see that these plants were viable. We didn't have major growth defect, were producing flowers and seeds without any problem. So what I did mainly was to do ionomic analysis. Ionomic corresponds to overall analysis of the ion profile of different elements in different mutant backgrounds. And here I'm showing you the leaf ionome in a Casparian strip mutant compared to a leaf ionome of a Suberin deficient line. The data are presented in the level are presented in percentage compared to the wild type levels. If we are in the yellow zone, that means that the element accumulate at higher level in the mutant compared to the wild type. If we are in the blue zone, that means that the element accumulate at lower level in the mutant compared to the wild type. And when we did these ionomics, we were very surprised for a while because we were really expecting that if we had mutant without Casparian strip or without Suberin, we will see a massive overaccumulation of many elements, and this is not the case. Most elements are not affected in these lines or mutants. We can see some that overaccumulate, like magnesium for the Casparian strip mutant or sodium for the Suberin deficient lines, but this is not the general trend. On the contrary, we actually see some elements that accumulate at lower level. This is particularly the case for potassium, where we have a decrease by 40% in a Casparian strip mutant and around 20% in a Suberin deficient lines. And I'm showing you this result because, yeah, it was not what we conceived at first, but this illustrates a fundamental but often overlooked property of the endodermis as a bidirectional barrier. So we all learn in plant biology that these barriers play a very key role to filter what's entering in the plant and can protect the plant for excess uptake of some elements that can be sometimes toxic. But if this is true, they can also potentially block the leakage of some element that would be accumulated by the plant and present in the vasculature. And we think that this is the case, especially for potassium, where we see that when we have no functional Casparian strip or no Suberin lamella, plants accumulate much less potassium. And we really think that potassium is actually not kept as much as it could be when the barriers are functional, when these barriers are deficient. Anyway, so now I'm an assistant professor in Geneva, and I still work on the radial transport of nutrients in root. And so what my lab aims to do is to get an integrative view of these different pathways, studying them one by one and see how they are interconnected. So current projects in my lab include work on the symplastic pathway itself. We are also working on nutrient carriers. And of course, we also do a lot of projects concerning the Suberin lamella. And this is what I want to tell you a bit more about now in the coming minutes. So Suberin lamella. So I already said that Suberin deposits in the endodermis after Casparian strip are formed. Actually, Suberin has a very interesting pattern of deposition in the root because first it starts to deposit it as patch of endodermal cells that are suberized before a stage where we have actually a continuous suberization where the whole endodermis is suberized. So there are different ways that we are using to visualize suberization in root. So our favorite one is Suberin staining with fluoral yellow, where we can really whole mount stain roots, and then we can visualize Suberin in entire plants or roots. And we also have a Suberin biosynthesis reporter line where we use the promoter of GPET5, a key gene for Suberin biosynthesis. And we can then visualize with live imaging where is Suberin biosynthesis happening in roots. And we have really exactly the same pattern with this marker because we again see, first we have patch of endodermal cells where there is Suberin biosynthesis, and later on the whole endodermis does Suberin biosynthesis. Now in my lab, we are more and more analyzing the Suberin with fluoral yellow staining on wall mount seedlings. So we do some tiling microscopy where every tile is at very high resolution, and we can reconstitute the whole plant, which means that we can have an overview, but we can also zoom in our picture and get really information about the patterning, the cellular level. And this allows us to do semi-high throughput quantification of suberin deposition along the root. So we can see that we can quantify the zone of the root that is non-suberized, the zone that is called patchy, and the zone that is continuous. And this is very convenient when we want to compare mutants, condition, and so on. And you will see some of my data where this is very useful. So this pattern is what happened in the Arabidopsis plant, wild type and stress. We will see later in my talk that there are many reasons for this pattern to be changed. Stress can affect this patterning, but we also know a lot of mutants that can affect this patterning. So now if we go a bit more a step back in terms of suberin, not looking necessarily in the endodermis, but what we know about suberin biosynthesis and deposition in the apoplast. So we know a lot about the suberin biosynthesis itself. So many genes involved in the fatty acid biosynthetic pathway, phenylpropanoid pathway were studied. And we know that what's happening is that inside the cell, mainly associated with the endoplasmic reticulum, there will be production of suberin monomers. And these monomers need to be exported from the cell to be polymerized into suberin and then deposited at suberin lamella between the plasma membrane and the primary cell wall. How these monomers are exported from the cell is not really well understood. So many work focused on ABCG transporters, and it seems that they play an important role in the process, but they are not really the key mechanism, these three transporters, that can really export these suberin monomers. Secretion was suggested for a long time to be important in the process, but was not really studied in details, especially because it was really difficult topic to study. So, and I'm going to show you some of our recent data that support a key role of secretion in deposition of suberin outside the cell. This started when we worked with Casparian strip mutants, and we look at them at the ultrastructural level, and we could see accumulation of big vesicles at the periphery of endodermal cells. So here I'm showing you the Lord of the Ring 2 mutant, that is a well-known Casparian strip mutant. You have two endodermal cells next to each other. And we see here, attached to the plasma membrane, we have these big vesicles that are around 500 nanometers wide, so they are pretty big. And inside, there are a lot of internal structures. So we wonder what was happening in this mutant, if it was due to this Casparian strip defect or maybe other aspects. And we've actually found that many of our Casparian strip mutants were having the same structures accumulating in endodermal cells. So here you have the ESB1 mutant where we see the same vesicle. We have the Cas1, Cas3 mutant where we see again the same vesicles. And these vesicles, we could observe them very close to the root tip. Here we are at 2 millimeters from the root tip at a position where we absolutely don't see them in wild type plant. So we then figure out that this was indeed associated with suberin, because if we look at the pattern of suberin along the root, these mutants that are Casparian strip mutant, what's happening is that they have an enhanced suberin phenotype. So suberin starts very close to the root tip in this mutant. This is a very well-known response in response to Casparian strip defect that involved the receptor kinase Schengen tree and its ligand, the CIF peptide. And what will happen is that when Casparian strip are defective, a signaling will be triggered that repair the apoplastic barrier, putting ectopic lignin, but also ectopic suberin very close to the root tip. So when we were at 2 millimeters from the root tip in this mutant, we were in the zone of suberin deposition. And we can see that in this ultrastructure analysis, where you see, especially in the Lord of the Ring 2 mutant, we have these big vesicles, and they are really clearly in front of the place where suberin is being deposited. We then tested that in other conditions, mutant, in wild type plant, when we have also the suberization happening. We also have a stress where we can induce suberization and we can again see these vesicles. So we could clearly demonstrate that these vesicles, they are constantly present in suberizing tissues. So when I say that we see a lot of these vesicles, here I'm showing you just one picture in one section of a portion of a plasma membrane. I want to show you what I mean when I say that we mean a lot of vesicule, with showing you a 3D reconstruction of one endodermal cell. So here in the upper part, you have one endodermal cell that is suberizing, and we're going to move in Z in this section, and you're going to see many of these big vesicles present at the periphery. So here you have one, here you have another, and so on. So now we are moving in Z. And then we will do the 3D reconstruction. So this is a cell suberizing. And then we see, like, if we do a 3D reconstruction, there is a very high density of this vesicule accumulating at the periphery of suberizing cells. So later we wonder, of course, what are these vesicles, you know? So when we look at them in 2 dimensions, we can see that they are clearly directly attached to the plasma membrane. So there is clearly a fusion with the plasma membrane. They are in front of Suberin lamella being deposited, but they also form of internal, they are constituted of internal, maybe vesicles inside. But of course, in 2 dimensions, we don't have a full picture of what is the internal structure. And then we did 3D reconstruction to understand if it was internally extracellular vesicule or maybe tubules. And when we did a 3D reconstruction of this internal structure, we could see that we have a highly reticulated network of something that we can call vesiculo-tubules. So this is what we could observe with chemical fixation. We wonder if maybe the chemical fixation itself could lead to some artifact explaining this high reticulated network. So we did some cryofixation, and we can still see that we have tubules that form a network, but with much lower density. You see that they are bigger. They really resemble like ginger root. And these structures we call extracellular vesiculo-tubular containing bodies. We constantly see them, again, even with the cryofixation, in cells that are suberizing. So there are a lot of things we still don't know about this work. So we don't know what is really the compartment that is involved here. Is it something derived from the ER? We like to think so because this biosynthesis occurs at the ER, but we don't have a proof yet at this stage. Could also be derived from the multivesicular body, especially in 2 dimensions. It really resembles multivesicular bodies, but the reticulating nature of the internal structure will go against this hypothesis. But as I say, we don't know yet. What we think at this stage is that this vesicule could carry directly some lipidic compounds, maybe some pre-mature suberin, because when we stain for suberin in root where we highly induce suberization, we can often see also some vesicles that I stain with the floral yellow that's specific for suberin. And the size of this vesicle that can be stained with floral yellow correspond to the size of the vesicle we see at the ultrastructural level. Unfortunately, there is no way we can correlate the 2 types of experiments, so this remains mainly hypothetical. What we know, though, is that if we block secretion, we block suberin deposition, so really supporting even further a key role of secretion in suberin deposition. So for this, what we do is that we can use ABA that was shown previously to induce ectopic suberization, especially in the cortex. So in white-eye plant, if you apply ABA, you can really ectopically produce suberin in the cortex, and then we can manipulate genetically or pharmacology secretion. So when we do that, and we use a specific background that is a BFA-sensitive gnome-like one, so that's a background where if you don't put BFA, it behaves like a white-eye plant, but if you apply BFA to the plant, secretion is blocked after the ER. So in this background, without BFA, when we don't induce the interference with secretion, we have suberin deposition in the cortex, but when we apply BFA, there is no deposition in the cortex. This is something we could confirm with quantification, and at the EM level, the BFA-sensitive gnome-like one, when we apply ABA together with BFA, there is no suberin deposition and there is no accumulation of these EVBs, these big vesicles. So clearly supporting that secretion is indeed very important for suberin deposition. But as I say, this is quite exciting because there are still a lot of things we don't know, like the compartment, the origin, and the dynamic. Another important project in my group concerns suberin plasticity. I already showed you with mutant for Casper sweep or with ABA that we have ways to manipulate suberization. So in my postdoc, I started to study that and found that actually a lot of abiotic stress can affect suberization, especially mineral deficiency could really modulate. There is a enhanced suberin or reduction of suberin. We can have an ectopic suberin deposition in some condition and contrarily a degradation in suberin in other conditions. In particular, ABA is key in the induction of suberin and can really induce ectopin suberization in the cortex, as I showed before, but also along the endodermis. There are many other factors affecting suberin. Every couple of months, someone publishes a new stress abiotic or biotic controlling suberin. But what I want to highlight here is again the response to Casparian strip defect. And one of the key effects when there is Casparian strip defect is that the receptor kinase Schengen3, when bind to its ligand CIF peptide, will trigger ectopic suberization as well. Which means that we have two independent ways to induce ectopic suberization very close to the root tip. And this is what I'm showing you here. When we apply ABA for a few hours to seedlings, we have an ectopic production of Suberin very close to the root tip. And if we apply the CIF peptide to trigger Schengen3 signaling, we also have an ectopic deposition of Suberin very close to the root tip. We don't know genetic, and we know that these two pathways are independent. So when I started my group in Geneva, we had this nice network of signalings controlling suberization, but we didn't know any molecular factors involved in the endodermis to control this suberization. So that was one of my main objectives, to identify regulators of Suberin. So we started with a gene candidate-based approach, trying to identify transcription factors that would be involved in the process. And by analyzing gene expression of different transcription factors, especially some MIP transcription factors that were highly suspected to be involved in the process before, I'm not going to show you the detail of that for the sake of time, we found that several of these MIP transcription factors were expressed at much higher level in response to ABA or CIF peptide when we look at their expression in roots. This is particularly the case for MIP41, but we also see the same trend for MIP53 and 93, and also to a lower extent, but still significant for MIP92. So we decided to study this transcription factor in more details. Very quickly, we could have some comfort in our project because we could see that MIP41 promoter is active in the same cell as the one that makes Suberin. So here in purple, you have the MIP41 promoter activity, and in green, you have the GPET5 for Suberin biosynthesis, and we have a perfect co-expression. If we look along the route, actually, it's slightly more distant. So MIP41 is in the endodermis, but start a little bit before you will have the Suberin biosynthesis reporter, which would fit actually with the role as MIP41 is actually controlling suberization. Importantly, when we apply ABA or CIF, the two conditions that induce ectopic Suberin very close to the root tip, we can see that MIP41 expression is now also ectopically expressed very close to the root tip. We look at the three other MIP transcription factors, and they are all expressed in the endodermis. They are all induced by ABA and CIF peptide. At different degrees. So for MIP53 and 92, it's mainly in terms of intensity, while with MIP93 then start to be expressed very close to the root tip. We could see by overexpressing them with a promoter that is specific for non-suberized endodermis, where we make the Casparian strip, so a rather young endodermis, that they are all sufficient to induce suberization. And when we mutated the four of them together, we could see that we lose suberization in non-tutic condition, and most of the response to AB and CIF is also lost. So now we have this framework where we have four transcription factors that are important to control suberization in response to ABA or the Schengen3 CIF signaling cascade. Of course, there are still a lot of things that we don't know. So we don't know what are the targets of this MIP transcription factor, what are the upstream or downstream regulators, if there is any hierarchy among them. And we actually also think that there are even more regulators that are needed to control suberization. And in just a few seconds before I finish, I will tell you some other things that we're doing to identify even more factors and to understand the function. So we have different screens going on in my lab. One of them is with natural variation. So we're screening for natural variation in Arabidopsis for Suberin deposition. So Jiangpu Han, who is in postdoc in my group, is doing this project. He characterized the Suberin pattern in 284 accessions. You could see that there is a very high degree of variation for Suberin. Here they are ranked based on the continuous zone of suberization. So on the right side, you have plants with very enhanced Suberin deposition compared to on the left side, plants with reduced suberization. So we now have enhanced accessions that have very weak Suberin deposition or some with very strong Suberin deposition. And we are doing a lot of physiological analysis with these plants to actually understand the function of Suberin in plant adaptation. We also ran a GWAS analysis with this data set. We could see that there is a lot of genetic association with this Suberin deposition. However, none of them is very strong. So it seems that the polymorphism that affects suberization didn't structure Arabidopsis population. One of them contained MIP41, which suggests that this approach could be a good strategy to identify genes involved in suberization. And we are now characterizing loss of function mutant for some of the gene candidates from that. And we already have new mutants with defect in Suberin, but I'm not going to talk about that now. So to finish, so in my group, we're trying to really get an overview of the radial transport of nutrients in roots and the different pathways. So I told you what we do about Suberin, but this is still a work in progress. As I said, we have many screens ongoing where we want to identify new factors controlling Suberin and also understand better the function of this barrier. We also have projects concerning the nutrient carrier, the symplastic pathway, and we also want to understand how these different pathways are integrated. So it's quite an exciting time for us in Geneva, and I hope that if I see any of you in coming years in conference, we'll show you some updates on the other pathway. So with this, I'm done with my talk. I just want to thank my group in Geneva. I have a very nice group with people who joined me where I was just starting. So that's really want to thank them for joining me and working with me. I particularly want to highlight Vinay Shukla, who was working on the MIP transcription factor and is now doing a postdoc with Francesco Licausi in Oxford. We had Francesco's talk yesterday. And Damien Debelis, who did all the beautiful EM analysis that I show you during my talk. And I thank you all for your attention.